Alright, in the first podcast we talked about DNA and how it was made up of nucleotides and how those nucleotides were important in specific orders and how that determined genes and which proteins would be made and how they bound together to form the double helix. In this podcast, we want to take it to the next level of how we're going to take that DNA and package it into a very small structure, the nucleus. And we're going to package it in such a way that it's usable. So let's get started with this discussion of chromosomes. And remember, DNA forms this double helix. And within this DNA, we have specific genes. They would take up more than that small piece of DNA, but this is just representative drawing. But we might have several genes along a stretch of DNA. And as we talked about with the central dogma, most genes get converted into proteins. Some genes get converted into RNA that are never translated because their role is just to make RNA. But let's just talk about it forming this protein in this example now. So as this DNA is structured here, enzymes that are necessary to convert this gene into a messenger RNA so that it can be made into a protein have easy access to the gene to transcribe it. However, we don't want every gene turned on all the time. We want some genes to be turned off. So we have a couple needs I want to talk about of the cell. The first is the need for gene expression within the cell. How can we have a system where we have this large piece of DNA that's very open and accessible to enzymes? How can we make sure they're not all turned on at the same time in the same cell? If you think about a multicellular organism, we have many different kinds of tissues, and we have a need of those tissues, say in the pancreas to turn on pancreatic genes and in the liver to turn on liver genes, and not vice versa. So there has to be this level of gene regulation. The second thing we have to think about and be concerned about is packaging. Let me draw that problem over here. If this is a cell here, and inside we have a nucleus. A typical mammalian cell has a nucleus that's about 6 micrometers. The amount of DNA that has to get placed into this 6 micrometers diameter of a nucleus is 2 meters. Somehow we have to find a way to effectively put 2 meters of DNA here into a 6 micrometer nucleus. Not only do we have to package it in here, but we have to package it into this nucleus in such a way that it can be used. It's not like when I'm packing for a vacation, I just throw all my clothes in a suitcase and shove the door shut so that I can get on with my trip. And then when I get there, I can't use any of my clothes. Luckily, our DNA packs the nucleus in a much more usable manner. It puts the DNA in here so it can be used. But it's an extraordinary packing task it has ahead of it. So here I want to write packaging DNA. Let me put lots of DNA into a small nucleus so it can still be used. Let's spend a little time talking about how it accomplishes this feat. And before we get started with that, I think it's appropriate to talk about when we need to have it packaged up into a very discrete chromosome and when we don't need it as tightly packaged. It always has to be somewhat packaged. But let's think about the cell cycle. And we haven't really spent time on the cell cycle, so I'm going to review it with you just real quick, but rest assured we'll spend much more time talking about it. During G1, S, and G2, we call that interphase. This is a time when the cell grows, divides its DNA so it can enter mitosis where it segregates the DNA. In general, during this working phase of the cell cycle, when the DNA is being transcribed to make messenger RNAs to do the business of the cell, and during this time when it copies the cell, the DNA is what we would call chromatin, sometimes referred to as beads on a string, and I'll explain that in a moment. It's less packaged than the chromosome. During mitosis, essentially gene activity is absent. The genes have been shut down. The whole point at this point is to make sure that the two cells that are being produced have the same number of chromosomes. And a way to effectively do that is to condense this chromatin down to a highly condensed form of the DNA called chromosome. Now both of these, I should mention, chromosomes and chromatin, contain proteins. In fact, a lot of proteins. Proteins are very important in packaging either the chromatin or the chromosome. And the more and more we learn about all the different things that RNA do, we know that RNA is also associated with the chromosome and the chromatin. So I want to draw on the right side of this board. On the right half of this board, I want to draw a little bit of how the DNA is packaged. And at the most basic level, the secondary structure level here, we just have the double helix. It then gets packaged to this whole beads on a string, the chromatin. Okay, so now as we form this chromatin, what happens is this DNA becomes a little bit more condensed. I'll try to show it like so. And it does that by these proteins binding to it. These proteins with the DNA that are around it is called the nucleosome. And the nucleosome, as I just stated, contains these proteins, and these proteins, I should identify those are called histone proteins. Don't worry about the kinds of histone proteins and how they're held together. You'll learn that in future classes. So let me go ahead and identify what this structure is. It contains DNA plus histones. Histones are the proteins I drew here in red. The DNA plus these proteins together make this structure called a nucleosome. These nucleosomes will appear repeated throughout a piece of chromatin. So double helix to the chromatin. Before, I said that this is often called beads on a string. And the reason for that is because if you look at an electron micrograph image of chromatin, what you will see is the DNA appearing as the string and these nucleosomes popping out and looking like they're beads. Now your book, as most cell biology books do, and genetics books do, will go into an elaborate stepwise procedure of how this is condensed further and further and further until you ultimately get to a highly condensed DNA structure, which we will call the chromosome. And I'm going to draw 3 arrows here just to indicate that it doesn't go straight from the nucleosome, the chromatin, right into this step here, the chromosome. We don't need to worry about all these steps. And to be honest with you, scientists still don't quite understand how it goes to this step. And these different intermediates. We have some information about it, and we are slowly beginning to learn more about it, but it's still a mystery. So a lot of job opportunities in here for you. What I want to do now is draw a chromosome in a little bit more detail and identify some of the structures of that chromosome. So I'm going to draw a chromosome like so. And in humans and all diploid organisms, we have 2 copies of every chromosome. These 2 chromosomes that we have are called homologs. Homo for same. It's the exact same order of genes on each of these 2 chromosomes. They're each 2 separate chromosomes, but the order of the genes are the same. I want to identify some parts for you. On these very ends here are telomeres. Every linear chromosome on their ends are structures called telomeres. Telomeres do a few things, but for now, let's just say they maintain the chromosome's structural integrity. They ultimately decide how long this chromosome and the cell that it is in will exist. Let's point out this part here and this part here. These are called centromeres. These serve as attachment sites for spindle fibers, which are a type of cytoskeletal element that assists in separating the chromosomes during the cell cycle. Associated at these centromeres, and I'm just going to draw a circle here, and these are proteins called the kinetochore. Again, these are proteins that associate with the centromere that aid in the binding of the spindle fibers. The last thing I want to point out here are origins of replication. And there's not a specific site to find them, so I'm just going to do this. Eukaryotic DNA is very large, and being linear, it creates some problems when you replicate it. But you have to take genetics to understand those problems. It's more of a genetic question than it is a cell biology question. But what I do want you to know is as this DNA is being copied, it does so at multiple sites. So this brown color here are these origins of replication. So you should know there are several sites that are the origins of replication. It's not like the telomeres where you could draw it clearly, or the centromeres. Now if you remember when I drew that cell cycle, and let me just draw it here, that replication, when I'm talking about replication, that occurs here in the S phase. And what happens when it's replicated, I'm not going to draw it in the exact level of detail here, but remember earlier I said it does so in a semi-conservative mechanism. And so you get one original strand with one new strand. And we'll get two of them, right? Because we had two here. And they're held together at this centromere. So this is what it would look like at this stage of the cell cycle. Now what happens next, I'm going to hold off until we talk about the cell cycle later in the semester. But for now you should know that they enter M phase here, mitosis like this, and during mitosis these chromosomes are going to get pulled apart. But you'll have to wait till the end of the semester to know the rest of that story. I'll keep you hanging for just a little bit. So we've talked about how the DNA gets packaged into these chromosomes. The next thing I want to talk about is how are these chromosomes, these massive chromosomes, positioned in the nucleus. So if this is our cell here, and this is our nucleus here, how is it do we get, say in humans, 46 chromosomes? Humans have 46 chromosomes. We could also say humans have 23 pairs of chromosomes. Both would be the correct way to say that. Oh, and I should clarify, this is in somatic cells. All of our cells except our gametes. Our gametes, eggs and sperm, would just have 23 chromosomes. But our gametes always have half the number of our original chromosomes. But let's think about these 46 chromosomes right now. How do we get these 46 chromosomes in here? Just like the DNA has to be packaged in some usable manner, we can't just take all this DNA and cram it into this tiny little nucleus and expect to be able to use it. Rather, each of these chromosomes, and I always find this really impressive, but I'm kind of a nerd, so I find a lot of these things impressive. But these chromosomes, each of these 46 chromosomes, have a distinct place where they're at. I'm not going to draw all 46, but I do want to draw a few. Okay, I'm just going to stop at 6. But these would fill the nucleus. And these we call, these distinct regions here, chromosome territories. We're slowly understanding why chromosomes are positioned in certain places, how they're attached to the nuclear envelope, the importance of attachment to the nuclear envelope. There are specific diseases that are linked to the inability of chromosomes to specifically bind to this nuclear envelope. But one key feature of these chromosome territories is that they are important in gene regulation. There has to be a high-level organization within the nucleus so that the right genes can be turned on at the right time. And these chromosome territories help accomplish that feat. Now within these chromosome territories during the interphase, remember I'm calling them chromosome territories, but during interphase this is really that beads on a string, that chromatin structure. And we have, if I could expand one of these, I'll expand this one here because I have a little bit more room over here to talk about it. Chromatin within these regions can be of one of two types. It can be euchromatin or it can be heterochromatin. It's really important to understand these when we talk about gene regulation. So talking about these two terms in great detail really is better suited for genetics. So I'm not going to talk about them in great detail right now, but what I do want you to know, because they are part of this elaborate packaging of the cell, I do want you to know that euchromatin is relaxed and active. less packaged with those nucleosomes, it makes the DNA more accessible to enzymes that need to turn on these genes. Heterochromatin is heavily packaged still, so heavily packed, and represent areas that are inactive. So either genes that have been turned off, or there are large parts of the chromosomes that are just never expressed. Those would be heterochromatin. So euchromatin, active areas of DNA. Heterochromatin, low activity of DNA. These are positioned, these chromatin are positioned in these territories in such a way that the euchromatin can be expressed and the heterochromatin is repressed. Okay, that's all I'm going to say right now about DNA and chromosomes. If you have any questions at all over these past 2 podcasts, please make sure you come see me. If not, I'll see you in class.